Look at this image again. Next, I'm going to show you a histological section of the spinal cord passing through this region here. And so I will show you the spinal cord and parts of the anterior and posterior root, including the spinal ganglion, and a small bit of the spinal nerve. I think it's worth you observing how neurons and glial cells of the spinal cord appear with conventional hematoxylin and eosin staining that you are probably used to viewing in your histology laboratory classes. This section is not quite in the best orientation, but just tilt your head to the right and look at it. The white matter is here, and the grey matter is here. Here is the dorsal horn and the dorsal root. Further along is the dorsal root or spinal ganglion. You're probably wondering how I can tell dorsal from ventral. Ventral horns are larger, and see this little indentation here? That's part of the ventral fissure. So here is the ventral root. Then both the dorsal and ventral roots unite beyond or distal to the dorsal root ganglion to form the spinal nerve. Something worth noting in this image is that all three meningeal layers cover the spinal cord and extend over all the dorsal root ganglia. This outermost thick, dense, fibrous layer is the dura mater. This subdural space is an artifact. Here is the thin arachnoid layer and the underlying subarachnoid space. The pia mater is attached to the outer surface of the spinal cord. Beyond the dorsal root ganglion, the dura will become continuous with the epineurium of the spinal nerve. Motor neurons in the ventral horn are well preserved with a pale nucleus and a prominent nucleolus, and Nistel substance occupying much of the cytoplasm, as you saw in the previous section of the spinal cord. Again, about all that you can see of glial cells are their nuclei and a region that stains paler than the surrounding tissue, or a tiny clear rim of what would have been cytoplasm. And remember from the earlier section of the spinal cord that I showed you that the larger, paler nuclei belong to astrocytes, and they're the most numerous of the glial cells. The small, dense, rounded nuclei are oligodendrocytes, and the elongated nuclei belong to microglia, or maybe endothelial cells. These profiles here are the nerve rootlets running in the subarachnoid space. Nerve fibres emerge from the dorsal horn and the ventral horn all along the spinal cord. All the emerging fibres posterior to each vertebra collect together to form a pair of spinal nerves, left and right. To do so, some fibres must run for a short distance up or down along the cord after they emerge, and as they do so, they collect into bundles called rootlets, which, as you know, finally come together as two roots, a dorsal root and a ventral root. All along here, you can see these rootlets sectioned transversely running along the surface of the cord. This section of the spinal cord has well-preserved white matter as well as grey matter. Nonetheless, myelin sheaths have been extracted and the axons are considerably shriveled in the spinal cord. It's very hard to avoid this sort of artifact, which affects the large axons more than the little ones. Myelin is more easily preserved in the nerve rootlets than that within the white matter of the spinal cord. Now look at these superbly preserved axons running in bundles, or the nerve rootlets, just outside the spinal cord. they show that axons actually are quite plump structures. But let me warn you that you may gain a different impression when you come to look at shriveled axons in peripheral nerves. Because the preservation is so good, you also get an extraordinary view of the ependymal cells lining the central canal in this section. You can almost imagine cilia and microvilli present on their apical surfaces. The microvilli specialised to absorb cerebrospinal fluid. They are joined at the apical borders by typical junctional complexes you see in other epithelia, but there is no basal lamina supporting them. Look carefully here and here and note that the basal ends of the ependymal cells are elongated and extend and apparently branch into the nearby neuropil. Here is a cross-section of the spinal cord that has been stained with 2 silver procedures. Just as a review exercise, can you immediately identify white matter and grey matter, the ventral horn and dorsal horn, and here is the anterior or ventral median fissure. Look at all the brown stained nerve fibres extending between the grey and white matter. And out here are sections through nerve rootlets. This image at higher magnification shows part of the great complexity of neuronal processes that make up the neuropil. That is, the area where numerous synaptic connections occur, almost always in the grey matter. I can trace short segments of axons and dendrites here and here, then associate them with cell bodies in this preparation. Have a look at the white matter here, in this same histological section. The axons in it run primarily up and down the spinal cord, and so are cut transversely. Bodian stain is a great stain for distinguishing myelinated from non-myelinated axons. Some regions of white matter are made up primarily of myelinated axons. Others have mainly non-myelinated axons. And here I'm showing you some of the nerve rootlets on this section. You can't miss the difference between clumps of non-myelinated axons and axons with myelin.